So friends, the next technique under the lecture series of plant tissue culture is organogenesis and somatic embryogenesis. Now the practice of using the cuttings of the plant to grow newer plants is one of the oldest technique we can say that is used to propagate the plants, and therefore we can say that organogenesis is nothing but it is one of the oldest technique of asexual plant propagation that is largely done from the non-meristematic or the somatic tissues. Now it all initiated with Haberland, who is known as father of plant tissue culture, who gave the hypothesis of totipotency and stated that we can develop new plants from the cells of the plants. But it was in 1957 when the famous Skoog and Miller experiment showed that by changing or by maintaining the proper auxin to cytokinin ratio in the tissues, we can develop newer plants from any somatic cell. And in 1966, Torre, he advanced the theory of organogenesis after Skoog and Miller experiment, and he stated that in callus it starts with the development of a group of meristematic cells which we call them as meristemoids, and these meristemoids can lead or can get converted into primordium cells which lead to the development of either shoot or root, and this development or change or transformation of meristemoid into primordium is largely dependent upon the conditions or the factors that are to which the cells in the culture are exposed. So we can define organogenesis that it is an ability of the plant tissues to form various organs in de novo manner, or new organs are formed from the plant tissues. It is also defined as an ability of non-meristematic tissue to form organ de novo. Another definition states that organogenesis is nothing but development of adventitious organs or primordial from undifferentiated cell mass by a process we call it as differentiation. So here we can see that the cells, they undergo undifferentiation state, and after undifferentiation they again enter into differentiation to develop into newer organs of the plant. Organogenesis is also defined as formation of roots, shoot, flower, buds from the cell in culture under in vitro conditions. So again, if we look into all these definitions, one thing is common, that we can develop newer plant organs by using somatic or non-meristematic cells if we provide proper in vitro conditions in terms of the nutrient media, in terms of light, in terms of temperature or pH of the media. Now if we talk about developmental process, now developmental process is nothing but it is a process by which certain changes occur in an organism which makes it suitable for developing certain food or to reproduce or to grow. And therefore we can say that this kind of process happens both in plant cells as well as in animal cells, but there is a difference between the developmental processes that occur inside the plant in comparison to the animal cells, or we can say that developmental process in some ways is unique to the plants. Now how it is unique? Because if animal cells undergo developmental path or developmental process, they differentiate into specific tissues which are responsible for a specific function. Now this kind of differentiation in animal cells is quite irreversible, so they cannot go back to their undifferentiated state, whereas unlike animal cells, plant cells retain their ability to de-differentiate from their current structural or differentiated state. So once plant cells, they differentiate into a specific tissue which have a specific functional state, plant cells retain their ability to go back to the de-differentiated state. So this is the major difference between the developmental process of a plant cell and an animal cell. Now, if we look into the organogenesis in the plant cell, it has two major steps. One is calogenesis and second one is rhizogenesis. So, calogenesis is nothing but it is a process of formation of adventitious shoot system from the cell under the in vitro condition, whereas rhizogenesis is nothing but the formation of root cells or rooting of the cells under the suitable in vitro conditions. Now, this kind of calogenesis or rhizogenesis can occur directly from the explant or from the undifferentiated mass of the callus cells, which we are going to discuss ahead. Now, as we have discussed that organogenesis can directly can occur from the explant material or from the callus also. So, if we look into the stages of organogenesis, we can say that organogenesis occur or occur through mainly two stages. First one is dedifferentiation and second one is redifferentiation. So, as you can see in this image, we have a leaf which acted as an explant material. It is fully differentiated, the cells are fully differentiated, tissues are fully differentiated in this explant. We take the explant onto the culture media, thereby we grow an adventitious shoot system or adventitious root system. Now, before that growth, the explant cells undergo dedifferentiation. They will lose their specific functionality which was occurring in the explant. So, they undergo dedifferentiation, and by exposing them to suitable cultural condition, we can introduce redifferentiation into those cells, by which we can induce organogenesis. And once organogenesis is introduced, proper differentiation of the cells occur and we get newer plantlets through this whole process. So, we can say organogenesis occur through dedifferentiation first and again redifferentiation of those dedifferentiated cells. Now, if we look into the product which is obtained after organogenesis, we can have three types of organogenetic products. One is shoot regeneration, wherein we obtain adventitious shoots from the explant material or the callus material, and that is why it is known as shoot regeneration. Since only shoot is obtained through this, it is a monopolar sort of regeneration process. Second is root regeneration, wherein we expose the explant or the callus cells to specific conditions which favors the regeneration of roots only. So, therefore, it is also a monopolar mode of development. And third one is the somatic embryogenesis, which can lead to both formation of shoot as well as root system, and therefore, it is largely considered as a bipolar mode of organogenesis. The two major modes or types of organogenesis is indirect organogenesis or de novo organogenesis, and the second one is direct organogenesis. Now, as the name indirect indicates, it occurs through the callus cells. So, the differentiation from the dedifferentiation of an explant leads to formation of callus. Callus is subjected to the specific growth conditions, in vitro conditions, which lead to the formation of meristemoids, and through these meristemoids new organs of plants are obtained. So, since this growth or organogenesis is occurring through callus, we call it as indirect organogenesis. Now, if we are subjecting that callus for we are subjecting these callus cells to subculturing processes for a very long duration, then such kind of callus cells may lead to somaclonal variations. There might be certain morphological or genetic variations may occur in the meristemoids or the newer plants which we are getting through the indirect organogenesis. On the contrary, direct organogenesis is a technique wherein shooting or rooting directly occurs on the explant and no callus is involved. So, explant material is exposed to specific culture conditions wherein the cells in the explant, they undergo dedifferentiation and they develop into meristemoid. So, no callus is involved, all such changes happens on the explant material itself. And since no callus is involved, subculturing does not occur and there are very less chances of somaclonal variation. And this technique of direct organogenesis has been found more suitable for herbaceous plants. Now, on this basis of dedifferentiation and redifferentiation, Hicks proposed a model for organogenesis by breaking different processes or different phases of organogenesis into different subphases. So, here you can see that X plant first undergo dedifferentiation, where the cells, they lose their specific functionality of that X plant and they form undifferentiated mass of the cells. Once they enter into dedifferentiation phase, now second step is to attain the competence to get converted into the to enter into the redifferentiation phase. So, competence is, we can say that, is an in between stage of dedifferentiation and induction. So, once the cells achieve the competence or they are competent enough to switch into the induction phase. Now, induction is the stage wherein the cells become ready to redifferentiate. So, after induction, it has been observed that the cells should be determined to get redifferentiation, and once they are determined, they enter into differentiation phase and the organ is developed. Now, we will discuss all these three stages: dedifferentiation, induction and differentiation. So, dedifferentiation, it involves the reversion to a less committed or more flexible or plastic developmental state that may or may not give rise to callus tissue. So, if it gives rise to callus, it becomes indirect organogenesis. If it occurs directly on the explant, it becomes direct organogenesis. So, in direct organogenesis, competent cells do not produce callus tissue and are thought to be the sole organ progenitors. They undergo the differentiation process and proceed individually or perhaps in small groups to produce new primordia, which act as the base cells for the development of organs. Then after dedifferentiation, they enter into induction. Now, this induction occurs between the time the tissue becomes competent and the time it becomes fully determined for the primordia production. So, as we discussed, once the cells get dedifferentiated, they have to gain the competency to induce the redifferentiation process. Now, this phase has different phenocritical times. Now, we know that the development of the plant cells into specific tissues with specific functionality follow a specific developmental process or the path. Now, this specific developmental process depends upon the predetermined genetic pathway in the normal developmental course of the plants. Now, during this developmental processes, there are certain specific time period when specific genes become active, which move the developmental path into the predestined process. Such specific time periods, we call them as phenocritical times. These phenocritical times help in advancing this developmental process and leads to the morphological differentiation due to integrated genetic pathway in the normal conditions in the plants. Under culture conditions, we provide certain chemical and physical stimuli, which we call them as phenocopying agents. Now, phenocopying agents are nothing but these are the chemical or physical stimuli which interrupt the normal genetic pathway, normal course of development at specific periods, which we call them as phenocritical time periods. So, these chemical and physical stimuli can interrupt, inhibit certain genetically determined developmental pathway, thereby modifying the morphogenetic results, or which leads to the production of mutant like phenotype or phenocopy. So, we can develop either shoot system or root system, thereby interrupting the predetermined genetical developmental pathway. Now, these phenocopying agents are effective on a specific time, which I have already discussed, and these critical time periods we call them as phenocritical times, which define key steps in the genetic pathway of the developmental process. Now, once this induction process is over, so the end of the induction process is basically the point when the cell or the group of cells become fully committed to the production of shoots or the roots. So, they are determined to get converted into the shoot system or the root system, and this is attained when the explant can be is removed from the media and is placed on a basal media which is without any kind of plant growth regulator. So the cells are now sufficient enough on themselves to develop into a shoot system or a root system, and when this happens we can say that tissue is now fully determined for redifferentiation either into shoot or into the root system. So this is how the organogenesis occurs or takes place. Now the extent of that explant tissue becoming competent and eventually determined is in part a function of the physical and chemical environment to which they have been exposed. So whatever conditions you are exposing, which you are maintaining in the culture media, that will define whether it develops into a shoot system or into a root system. The measure of the extent of a development commitment towards particular endpoint is known as canalization. So Waddington defined canalization as the property of developmental pathway of achieving a standard phenotype despite genetic or environmental disturbances. Now we move on to differentiation aspect. So we discussed dedifferentiation, next step is differentiation. Now in this phase, morphologically differentiation and development of the nascent organ begins. So once the tissues or cells become determinant at the end of induction phase, the next step is differentiation, where they differentiate into morphological organs. So organ initiation involves an abrupt shift in the polarity followed by smoothing of this shift into radially symmetrical organization and concurrent growth along the new axis to form the bulge characteristic of organogenesis. So once the cells are determined, they enter into differentiation phase. During differentiation phase, there is a change in the polarity of the growth of the cells. They start dividing and they divide along a new axis to form a bulge, which we call it as a bud. So this initiates the final organogenesis process, that is the bud formation. Now in this image you can see these are the scanning electron microscope images of an explant in the A image, which has not been exposed to any kind of shoot bud induction media. So you can clearly see the stomatal rows or the stomata cells are surrounded by subsidiary cells, and each row of stomata is separated by the longitudinal rectangular cells. So they have a specific arrangement. In the picture B, this picture B was taken 3 days after the exposure of the explant to the induction media. Here you can see the surface become more or less rough in nature. There is no specific arrangement of the cells, and the cells start they are ready to divide. And in the third image you can see that the rearrangement of the cells is now coming back into the picture. Cells are not now started dividing. And fourth is the formation of the bulge, that is the formation of bud, which will eventually lead to the formation of newer plant or organs. Another example is the bud formation in Pinus ocopara species, seedling explant, wherein you can see the meristematic domes in picture A after 2 to 3 weeks of exposure to the culture media. Second image shows differentiation of this meristematic dome into leaf primordia. The third image, C image, show further differentiation into the leaf primordia, and fourth is the complete formation of the bulge or the developed shoot bud from where the shoot system will develop. So this is how the organogenesis process takes place in the cultured cells. Now the process of root initiation, this was we discussed about the shoot initiation, but in case of root initiation there are 4 stages. First one is the formation of a meristematic locus by dedifferentiation of the stem cells or the cells. Second is the multiplication of these stem cells into spherical clusters. Third stage is the further these cells multiply with initiation of planar cell divisions to form a recognizable bilateral root meristem, and eventually in the fourth step cell elongation of these cells take place, which are located in the basal part of the developing meristem, resulting in eventual emergence of a newly formed root system. So by and large, if we compare shoot and the and the root development process is almost similar. The final outcomes are different. One leads to the formation of shoot and second leads to the formation of roots. Now move on to the factors that affect organogenesis. We have donor plant growth, ex-plant characters, media properties and environmental conditions. So coming on to the donor plant. So we know that from where we get the ex-plant or the donor plant, it should be healthy. It should be free from any kind of disease, because eventually it will develop into the cultured cells. So if the donor plant is diseased, we are not going to get good cultures. Age of donor plant is also an important factor while taking ex-plant from that particular plant, because younger the donor plant, better are the chances for developing successful cultures, because younger cells have more vitality, have more vigor for cell division, and they can withstand the stress that has been exposed during the culturing process. Second is the ex-plant characters. So under ex-plant characters, we have size of ex-plant. Larger the ex-plant, better are the chances for better culture development, because larger ex-plants have more parenchyma, more vascular tissue and cambium cells, which will help in proper cell divisions. Then source of the ex-plant. The source of the ex-plant largely varies from species to species. Some plant species develop better when leaf is used as an ex-plant. Some develop better when stem part is used as an ex-plant. So it depends from species to species. Now the genetic instability of the callus culture increases with the time in culture, or we expose those culture cells for subculturing. More subculturing we do, more genetic instability will be there, and therefore it is always recommended, if we are going for indirect organogenesis, we are using callus cells, then freshly or newly initiated callus cells should be used for organogenesis, because lesser genetic instability will be there in the fresh initiated callus cells. Age of ex-plant is another important factor that will determine whether the final culture would be successful or not. As a general rule, younger the ex-plant, better are the chances, because again younger cells have more vitality, more vigor to divide, and they can withstand the stress better. Nicotiana, I have taken an example of Nicotiana. The regeneration of the shoots from the leaf ex-plant was found better when the ex-plant was collected before flowering stage of the plant. Similarly, bulbs of Lilium species, when collected during spring and autumn seasons, they give better results than those collected in summer or winter. So age and time of collection of the ex-plant also plays an important role. Third is the media properties. What kind of media you are using. So concentration of growth hormones is critical. Type of the growth hormone you are using is again critical, and that will determine whether shoot is going to be formed or root is going to be formed. The concentration of growth hormones that you are going to use in the culture will be dependent upon the endogenous concentration of the hormones which are present in the plant cells. So again, that will again affect the organogenesis process. Increased phosphate or adenine levels in the media counteracts the inhibitory effect of auxin and promotes the bud formation in the absence of cytokinin. So this was a study wherein it was found that if we enhance the phosphate or adenine concentrations levels in the media, they inhibit the auxin effects even in the absence of cytokinin, and promotes the bud formation. Then pH of the media is another important factor, and it has been found that pH range of 5.6 to 5.8 was suitable for the development of organogenesis. Culturing done either in callus or suspension, subculturing should be done at appropriate time intervals, because as soon as the media get exhausted, the callus cells, whether on the solid media or in the liquid media, they start losing their vitality and vigor to further withstand the culture stress, and therefore subculturing at proper time intervals should be done. Environmental conditions also play a role in the success of organogenesis, and temperature is one of such factor. Optimum temperature is 25 degrees, that will help in proper organogenesis, but depends upon the ex-plant. Certain ex-plant like bulbs, they grow better in a temperature range of 15 to 18 degrees. Intensity, light intensity to which the culture is being exposed also help in determining the success of the culture. There should be an optimum intensity of the light, because a high intensity of light has been shown to be inhibitory for shoot and bud formation. Not only the intensity, but the type of the light also affects the culture outcome. If the culture is being exposed more towards the blue light region, it promotes shoot formation, whereas if it is exposed to red light region, it promotes root formation. Then gradient of oxygen, because cells are dividing in the culture, they require a large amount of oxygen for their growth. So lower oxygen gradient is there, it promotes bud formation, and if high oxygen gradient is there, it leads to the formation of root system in the organogenetic cultures. We also talk about endogenous factors, that is the amount of the hormones that is present within the cells. So if gibberellins are present within the cells, they suppress both root and shoot formation, and therefore one has to take care of the presence of gibberellins in the cells. Similarly, ethylene also, presence of ethylene also blocks the organogenesis, and it has been seen that during the development of the cells in the culture, ethylene content also increases in the media, and therefore it also becomes important for the subculturing, or to replace the media with the fresh media. Carbohydrate act as a source of energy, but also they are very good osmotic agent. So proper concentration of these carbohydrates should be there in media to maintain osmoregularity, because once there is any change in osmotic conditions of the media, it will lead to osmotic stress that will hamper the culture growth. So friends, this was about organogenesis, and in the next lecture we are going to start with somatic embryogenesis. Thank you.